We are approaching our scheduled time, so let's go ahead and get started. My name is Jessica Vionis, and I'm the Director of Marketing here at Golden Helix. Today, I am very excited to introduce Greta Linse Peterson, who is Golden Helix Director of Product Management and Quality. Greta's main focus is managing SNP Variation Suite from strategic planning to tactical activities, including defining market requirements and working with our developers to ensure timely releases. On the quality side, she's responsible for software testing, quality control, and maintaining technical documentation. In addition to those duties, Greta also manages Golden Helix technical support team. She writes Python scripts for extending SVS functionality and conducts software training for customers and prospects. So she is quite a busy girl. Greta joined Golden Helix in 2008 when she completed her master's degree in both mathematics and statistics at Montana State University in Bozeman. And so with that, I will pass it on over to her.
Thank you, Jessica.
Thank you.
If you have any questions during this webcast, please feel free to enter them into the questions pane in the GoToWebinar interface. We will try to answer as many of them as possible at the end of the presentation today. And with that, let's go ahead and get started because we have a lot to cover today. The focus today is on agrigenomics and research workflow, or advancing workflows in SVS. With that end, we are going to cover two different workflows, one for GWAS and one for DNA-seq. We will try to give about equal time to both of the workflows. However, the GWAS workflow is a little bit more complicated, so it's going to demand a little bit more time for that. The GWAS workflow will use the sheep HapMap data and a simulated phenotype. And for the DNA-seq workflow, we're going to go ahead and use 3 bovine samples, partial exome sequences, and actually 2 of the samples are bovine and 1 is bison aligned to the bovine array, or a bovine reference sequence. Our software is used in a variety of settings for agricultural research. Today we are going to be talking about 2 mammalian species, but we can use it as well for plant, and we have had a customer recently use it for parasite research as well. And here are just a few of the institutions that you might be familiar with that have used our software for their research. In case you're not familiar with Golden Helix's SVS, or SNP Variation Suite, was created specifically for genetic research, and it was initially developed in the GWAS era. The performance, usability, and visualization have made the software an obvious and popular choice for genetic researchers and has resulted in hundreds of citations in peer-reviewed articles. More recently, as the world of genetic research evolves, the software has added numerous tools to account for a variety of workflows. SVS now includes GWAS, CNV, PBAT, RNA, and DNA sequencing packages, as well as an incredible and intuitive visualization powered by GenomeBrowse. Golden Helix's GenomeBrowse is a free genome visualization tool that was first launched in 2011. It makes the process of exploring the coverage and pilot data from DNA and RNA sequence pipelines intuitive and powerful, especially when visualized in conjunction with VCF files in the case of DNA sequencing. All the genomes that SVS supports, GenomeBrowse also supports, which is to say more than 24 plant and animal genomes are currently available. In October of 2013, the SVS and GenomeBrowse technologies were brought together in one product to create SVS 8. The demonstrations today will use SVS 8. This release is available, but as a few features are still missing, it is not the default release. If you do not already have SVS 8, you can choose to upgrade to SVS 8 now, or wait until the official release due out in the first quarter of 2014. Again, SVS and GenomeBrowse currently support over 24 plant and animal genomes, and we are adding more each month. Priority is given to the genomes requested by you, our customers, and that have gene and reference sequence sources available. So if your sequence or species is not currently available, please just let us know and we will add it to our list and we will get it built and available in our software as soon as possible. Let's begin with a demonstration of the GWAS workflow in SVS 8. This workflow will be condensed as there is not enough time to cover all the possible quality assurance or analysis tools that are available in SVS today, but I hope it's a good overview of what SVS is capable of. First, a little bit about our data set. I first encountered the sheep HapMap data set when I was looking for good data to use for the mixed model webcast I did last year. And I will refer to this webcast a few times, and Jessica will make sure to put the link for the webcast in the chat pane of GoToWebinar. I'm going to use this data set again for this webcast, for the GWAS portion, because it is so diverse in the number of breeds in the data set and has a large number of samples. For this webcast, I use a little over 2,000 of the samples and simulated a new phenotype for this data to not, as the focus is more on the different types of analysis workflows and not as much on mixed models itself. Originally, this data was formatted in the Planck PedMap format, and that is easy to directly import into SVS. It already has the genomic position information contained in the map file and is applied to the spreadsheet on import. However, for this webcast, I converted the data into Impute2 output format, and some other companies call that the Oxford format, to demonstrate the ease of importing data from imputation programs into SVS. The phenotype, again, the simulated phenotype for today is going to be a post-weaning weight gain phenotype, and the distribution of this phenotype matches published phenotypes so that we have some realistic data to use for analysis. Some markers were also removed to have either by the consortium or because they were unmapped, and the one marker from chromosome Y was also removed. The GWAS workflow we're going to cover today is first we'll do a little bit of a quality assurance process. We'll check out the phenotype distribution to make sure that it's normally distributed without any outliers that need to be removed from the analysis. Then we will quickly examine sample quality, SNP quality, and population structure. For the analysis portion, today we will perform 3 different tests or analyses, and the correlation trend test, linear regression with PCA correction, and EMMA-X, one mixed linear model analysis. This will be used for demonstration purposes to compare the results from the 3 different methods. In the end, we should see that the EMMA-X method was the best one suited for the analysis of this data due to the large number of breeds and population structure. To make that determination, we will look at the QQ plots and the Manhattan plots for all 3 methods. As a statistician, I do have to remind you that in your own research, it is better to have a hypothesis going in that also includes which test you're going to perform. Theoretically, or technically rather, running 3 different tests or models would require adjusting the Bonferroni correction to account for all 3 of the different methods or models. In this case, we will look at them as we are doing this for demonstration purposes. If you do know that your data has a lot of population structure, cryptic relatedness, or inbred samples, EMMA-X is, I would like to argue, the best approach to go about analyzing that data. All right, let's go ahead and jump into our demonstration portion. Just flip over to SVS. And I already have my starter project ready. If you do not have a project created, one thing with the agrigenomics research is you do want to make sure that when you specify your new project that you go ahead and you change your default genome assembly. This will help make sure that the correct data or assembly is chosen for certain default annotation sources or parameters in the software, and as well as for visualization, your data will be visualized against the correct reference genome. And as you can see, we do have multiple species and multiple builds for each species. Since I already have a starter project created, we'll just go ahead and open that up. If you're not familiar with SVS, we refer to this as our project navigator. We do have a node changelog where we keep track of all of the changes that have been made to the project or to each node itself or spreadsheet. And we do allow you to take notes as well in the user notes panel. I already have the genotypes imported, but I will show you how I imported that data, and then we will import a few other spreadsheets. Again, this was imported from the Impute2 output format. And if we go to the import menu, we can select import to GWAS files. The Impute2 format is a paired format where you have one gen file that contains the genotypes, and you also have a sample file that contains the sample names as well as some missingness rates for each sample. We have one file per chromosome, and so we're just going to select all of those. We have the 26 chromosomes and chromosome X. It's white space delimited. Our files are prefixed with CHR. We will use the SNP ID for our marker names. And now we need to select the sample file. Again, because there is missingness data for each chromosome, we only need to, we do create one file per chromosome, but we only need one of the files to get the sample names. So select that. And I'm going to convert the dosage calls to genotype calls on import. You can also import the dosages as dosages to analyze them directly, and you can specify a new file name and click import. Again, I've already imported this data, so I'm going to go ahead and cancel out of this process, and we'll open up our spreadsheet. So in this spreadsheet, we do have the secondary ID column, which you do not need. So I'm going to double click on the column name header to turn that to gray, which means it's inactive. And I'm going to do the same thing for the missing value column. And then we have all of our genotype columns. We have about 48,000 genotypes for this data, and they are marker mapped. And starting with chromosome 1, we can check. We do have data for autosomes 1 through 26, as well as chromosome X. I'm just going to create a subset spreadsheet to remove those two inactive columns, so we have one spreadsheet that just has genotypes in it. Now let's go ahead and import our phenotype data. The phenotype data is in a text file, so we'll import text and select the phenotype data file. This is actually tab delimited, so I will select tab delimited, and we'll just shorten the name to phenotype. And again, the phenotype that we're going to consider today is post-weaning weight gain. To compute that value, we use the birth weight, weaning weight, and the 6-month weight. And then just for the sake of completeness, we also have the pre-weaning weight gain value. We will examine this data after we have all of our spreadsheets joined together, so I'm just going to go ahead and close this. Now let's import the pedigree file. The pedigree file I have saved as a text pedigree format. And so we'll go to Family Pedigree, Text Pedigree, and select the file. Even though this is a text file, it is actually comma delimited. And I do already have an identifier column in this data set, so we'll select column 1, shorten the name, and the gender is already encoded as 01 with missing as question mark. Same affection status is all missing. If you have gender is encoded as 012 or affection status is 012, you can select those options and it'll be recoded to the correct value on import. SVS uses a value of 1 for female and a value of 0 for male, and a value of 1 for affected and a value of 1 for unaffected. If you look at our pedigree spreadsheet, we can see that it has a pedigree spreadsheet icon as well as blue column headers to indicate that these are the standard pedigree columns. The family ID for this data is actually the breed name, and we have the patient ID. For most samples, the father ID and the mother ID is missing. We do have gender, and again, we have affection status is all missing. Starting from this spreadsheet, we'll first join with the phenotype, and we'll keep all of the defaults. And then from the pedigree and phenotype spreadsheet, we'll add the genotypes. and we'll shorten this. All right, so because of the way that we've joined the data, and actually always our pedigree columns will stay on the left, and then the next few columns that we have are the phenotype columns, and if we scroll a little bit more to the right, we see our genotype columns. Now that we have our data joined together, let's go back to our project navigator and mark this spreadsheet as a starting point for our analysis. So I just right-clicked on that node and selected a green flag so that I know that that means start. The next step I want to do is look at my phenotype itself to make sure that the distribution of this phenotype is what I expect or is normally distributed. To do that, I'm going to look at a histogram. So I'm going to right-click on the column header and select Plot Histogram. And I'm going to make this a little bit easier to see. And I'm going to increase the bin count. And it is fairly normally distributed. We do have one outlier, but it is not that far away from the mean of the distribution, and so I'm not going to be too concerned about that. More importantly, we do not have any negative values, and so all of our animals gained weight. And I do not have any values that are very, very large. The next thing I want to do is actually look at the samples themselves, and we'll perform some sample statistics. So we'll go to the genotype menu, genotype statistics by sample, and by default call rate and heterozygosity will be output. I'm also going to do some gender inference and X statistics, and we'll run that. And very quickly it's going to calculate the call rate for autosomes and the X chromosome, heterozygosity rates for all the autosomes, all the chromosomes, and the X chromosome itself. To look at call rate, I'm actually hoping that the call rates are all really good. So what I'm going to do is I'm just going to sort this in ascending order. And I see that the lowest call rate is 0.9896. And so I do not need to plot a histogram of this because I already know that all the call rates are very good. However, I do want to look at the heterozygosity rate. And we'll look at the heterozygosity rate from all of the autosomes. We'll look at a histogram of this. And there are a lot of heterozygous samples, but it's not too large. The distribution is fairly what we would expect, and so we can say that that's the good data here. The next thing I want to do is check out the make sure that the reported gender matches the inferred gender based on the heterozygosity rate of the X chromosome. To do that, I'm going to inactivate all the columns. And I'm just going to reactivate this last column, the binary inferred gender. And I'm going to join this to the pedigree. And again, the pedigree columns are always put to the right. So the inferred gender is going to be in column 7, and I'm going to go to select, compare and activate by column agreement, add the sex in the inferred gender column, and we'll just run it. And all the rows matched, so I do not have any genders that were reported incorrectly. So that concludes the sample statistics portion. All of the samples are behaving as we would expect. The next thing we want to look at is the marker statistics. So if we go to genotype statistics by marker, we'll go ahead and compute statistics for the X chromosome. And that warning is just to remind us that we do not have an X chromosome adjustment. We'll just look at the call rate and the allele frequencies. You can also compute the statistics for Hardy-Weinberg equilibrium, but in this case we'll just keep it simple and just look at call rate and minor allele frequency. This time I'm actually going to plot a histogram of the call rate. And let's make this a little larger, increase the bin count. All right, now the smallest call rate is a little bit less than 0.8, but it does seem to be an outlier. Let me zoom in a little bit more. And in fact, there are a few markers down here that have a call rate less than 0.95. In general, that is a pretty good call rate. However, since it's so far out of the normal or where most of the other markers are, I think we will go ahead and remove all markers that have a call rate of less than 0.95. For the minor allele frequency, let's just go ahead and sort that ascending order, and we can see that we do have 11 markers that are missing a minor allele altogether, so they're monomorphic. Those markers in general will be excluded from analysis anyway, so we will go ahead and just remove those to begin with. We do also have quite a few markers that have a low minor allele frequency. Since most GWAS statistics require that minor allele frequency be large enough to ensure that there are cell counts in every, if you're doing a contingency table analysis, that you have a large enough cell counts in each, or large enough counts in each cell. Low minor allele frequencies can cause problems with the distribution or the test. In general, we usually remove minor allele frequency of less than 5%. For this analysis, we'll be a little bit more stringent and we'll remove all markers that have a minor allele frequency less than 1%. To actually remove that data, we'll go back to our pedigree, phenotype, and genotype spreadsheet, and instead of just calculating the statistics, we'll actually perform genotype filtering by marker. And again, we'll go ahead and include the X chromosome. We'll drop if the call rate is less than 0.95, and we'll drop if the minor allele frequency is less than 10%. And very quickly, it will inactivate in those markers that fail those criteria. We get the same report out again, but we have the additional drop column for the filtering results. I'm going to create a column subset spreadsheet, and we'll rename this to indicate that it is filtered. And we'll go ahead and close that spreadsheet. The next step in the analysis is looking at the population structure of the data. Before we run principal components analysis, the next step is to perform LD pruning to remove all the markers that are in LD with at least one other marker. And this ensures that we have the cleanest, smallest data set possible with the least amount of redundant data. And before we do that, we also need to make sure that we remove the X chromosome, because we do not want to make sure that we have, we want to make sure that we do not have a gender bias in our principal components analysis. So I've removed that, and then for LD pruning, we go to the genotype quality assurance LD pruning dialog, and we'll go ahead and change the window increment to 10, and we'll be really stringent on the LD threshold and change that to 0.1. We'll go ahead and click OK to run. Now this does take a couple of minutes, which we do not have time for today, and so I already have a list of markers that I want to keep for PCA analysis in a separate spreadsheet. So I'm going to go ahead and cancel that and go back to my project navigator. The data set, or the spreadsheet that I have this information in, is actually saved in an Excel file. So I'm going to go to Import Third Party, Browse, and select Markers to use for PCA. And we'll go ahead and click Next and Next, and it'll import a spreadsheet that has some marker information in it, as well as all the marker names that I want to use. Now I want to apply this subset of data to my phenotype, pedigree, and genotype spreadsheet. So I'm going to go to Select and Apply Current Selection to Second Spreadsheet. I'm going to apply the filtered rows, or because the marker names are in rows, to the filtered pedigree, phenotype, and genotype spreadsheet. And that's going to leave about 30,000 markers. It does also remove the pedigree and phenotype columns. So I'm going to click once to activate the first column, and I'm going to scroll to the right and then shift-click, just like in Excel, to activate all of the rest of the phenotype and pedigree columns. And we'll go ahead and create a column subset spreadsheet. And we'll rename this LD pruned, or for PCA. Now, for PCA analysis, we'll go to the genotype menu and genotype principal component analysis. We'll just run all the defaults, just get the top 10 components. You can output the number of samples less one component. So if we have, if we go over to our spreadsheet, this tells us the number of active rows, or number of rows and number of columns in our data set. In the bottom is the number of active rows and active columns. So we can see that we have 2,124 samples. So we could output 2,123 principal components. It takes the same amount of time, but today we only really need the first 10 components, so that's all I'm going to calculate. I'm going to go ahead and click Run. Again, this is going to take just a couple minutes, so I'm just going to go ahead and import the data that I need. It's actually in a Golden Helix DSF file this time, and principalcomponents.dsf. Again, I have my 2,124 samples and 10 principal components. To examine this data and look at the population structure, we need to join this to the pedigree. So I'm going to go ahead and click on Join and select the pedigree. Click OK. Again, I have my pedigree columns on the left, and if I scroll over to the right, I have my eigenvalue columns. Now we're going to plot an XY scatter plot to look at the principal components. You could plot an N by N scatter plot where you look at the top 10 components compared to each other. However, because we have so many different breeds in this data set, if we click on the column header here and do a value count, we can see that we have an incredible number of breeds. This is too many different categories for the N by N principal component plot to color correctly. So we're just going to do an XY scatter plot this way. And one trick is that you can get a couple plots. If you plot the second component on the x-axis, you can plot the first component and the third component on the y-axis, and we'll get two different plots. Go ahead and close this and make this bigger. And you can see that we do have some structure going on here. What's more informative is when we color this by breed. So I'm going to select the first plot, and this is the second versus the first component. And I'm going to color this by family ID, which is, again, the breed information. And we'll do the same thing for the second versus third. And if we click on one of these data points, we can look down in the data console, and we can see that this is, because the breed is contained in the ID name itself, we can see that this is a Scottish breed, and another breed that's from the same region, and another German Texel breed. If we look over on this axis, we have an Australian breed, another Australian breed. If we look on this axis, we have a Bangladeshi breed, Sumatra, and we can see that the animals are clustering with their same breed group, and the breed groups are clustering with their same genotype or geographic location, and the same behavior is happening as well with the second versus the third components. So there is obviously some population structure that we do need to account for when we run our analysis association tests. Let's go ahead and get set up now for our association tests. What I want to actually do is take our pedigree, phenotype, and genotype spreadsheet and join the principal components to that spreadsheet so that we have one data set that we'll run all of our analysis off of. I'm going to start from the principal components spreadsheet and join it with the pedigree, phenotype, and genotypes filtered. This time I actually want to go back here. I want to use all of the markers, not just the LD prune ones, so I'm going to go to Select, Column, Activate All Columns. And now this is call rate and MAF filtered sheet 2. So go back to the principal components, and join that sheet too. Well, this time we'll create a spreadsheet as a child of the project root to get a clean view in our navigator. And I've got a lot of open spreadsheets. I'm going to pop back to the project navigator, click on window, and I'm going to close all my spreadsheets. Then we'll reopen that one that we just created. Okay, now I need to set my phenotype to dependent, so I'm going to scroll over to my post-weaning weight gain. And the first test I'm going to run is a correlation trend test. The naive approach, well, we'll just see how well it does. We'll go ahead and run it on the X chromosome, even though it doesn't adjust for it. We'll do, again, the correlation trend test. We'll output data for the PPQQ plots. We'll show an inflation factor, and we'll go ahead and uncheck call rate and allele frequencies. We've already looked at that. We don't need to look at it again. And we'll go ahead and click Run. And in a short order, we'll have our results. The first thing that I want to look at is the inflation factor. So I'm going to pop back to the project navigator. The inflation factor is saved in the node changelog. If we scroll all the way to the bottom for that spreadsheet and scroll over, we can see that our inflation factor is 2.1, which is very high. Again, that's due to that population structure. To confirm that, let's look at the QQ plot. So I'm going to do an XY scatter plot. Expected negative log 10 p-value versus the observed negative log 10 p-value. And we'll add a y equals x line for reference. And we can see, yes, this data is very inflated. All right, moving right on. Now let's do a linear regression test accounting for the principal components. To do that, I actually want to have the X chromosome adjustment. So I'm going to do this analysis through the mixed linear model analysis dialog. And I'm not actually going to run a mixed model at this time, but I will do linear regression. And I will correct for the hemizygous males. We'll select the sex column for the gender identification. And we'll add the eigenvalue columns. And to do that, I'm just going to put an EV in the search box that brings up all 10 columns. I'll select all, OK, and we'll let's make sure we get the output data for PPQQ plots and uncheck call rate and allele frequencies, and OK to run. Now this is going to take a little bit more time because it does have to scan all the markers first, correcting for or adjusting for missing values, and then after it finishes the scan, it will go through and perform a linear regression on each marker. For more information on the mixed model analysis or the different methods used in this, again, I would encourage you to watch the mixed model webcast from last year. And very soon we will have out a mixed model analysis tutorial. It's just a little bit delayed with the waiting for the official release of our next version of our software. And almost done with the scan, and then we will move on to the regression analysis portion. This is going to run a little bit faster. This method can also be used for a binary phenotype. According to the Kong 2008 Emma X paper, this is similar to an arbitrage trend test. And so this is, even though it is a linear model, it is still, as long as cases and controls are balanced, it can be used for a binary variable, although it is linear is more associated with, when we think about it, with a quantitative phenotype. All right, here's our results. Again, the first thing we want to look at is the XY scatter plot. And so I'm going to plot the expected negative log 10p versus the observed, and add the y equals x line for reference. And this is a lot better than the naive correlation trend test, but it is still inflated. How inflated? Well, we can take the median of this distribution and compare the median of the observed versus the median of the expected. To do that, we have an add-on script. Add-on scripts are free Python scripts utilities that can be added on very easily to our software. To obtain add-on scripts, if you have not done this before, if you're new to SVS, you can go back to the Project Navigator, go to the Resources menu, and click on Add-on Scripts, and that will jump out to a web page on our software, or for our company. And you can download any of those scripts, and it provides directions on how to use it, how to install it. And once it's installed in the software, it's used just in the same manner as any of our other tools. So I'm going to click on the column number header for the p-value column and select one of these add-on scripts. It's called calculate pseudo lambda. And you see that our pseudo lambda, because it's not directly calculated off of the chi-squared, that's why we call it pseudo, it's 1.09, which is a lot better than the 2.1. And we could see that if we can get this a little bit better by the next approach. What this is going to tell us is that our data is still going to be a little inflated, but it's going to have a lot less false positives in this analysis than with a correlation trend test. The last method we're going to run is the EMMA-X mixed model. And to do that, we need a kinship matrix that identifies the relationship between all of the samples. You can go ahead and compute the kinship matrix at the same time that you're running this. However, just like with PCA, we recommend that you use only common, LD pruned, non or autosomal markers for that. And it takes just a few minutes to run. So again, I'm going to import that data already calculated. It is also a GSF file, and we'll import that and be able to use it. The nice thing about this is that you can create this once for your larger data set, and you can create subsets of this data and reuse this IBS matrix for all of your analyses. As long as the samples and the genotypes do not change, this information about relatedness will not change for these samples. So it can be reused for multiple different phenotypes, multiple different approaches. So let's go back to the spreadsheet, and we will run our EMMA-X analysis. Scroll down to grab that IBS matrix. Identity by state is a relationship, or how similar, it's a similarity matrix to tell you how similar the samples are. It's not the same thing as identity by state, where you would expect more values closer to 1 to be identical, values close to 0.5 being siblings. In this case, we're not going to correct for any additional covariates. We do want to output the data for the PPQQ plots and uncheck those. We'll go ahead and say run. You can use an IBD, identity by descent matrix, instead of an IBS matrix. And you can also use an explicit pedigree matrix if you have a clean data set that does not have a lot of inbred samples. However, that matrix itself would be a little bit time consuming to create. It's a little bit easier to just run identity by state. We also have another tool called GWAS, which some of you might be familiar with. GWAS calculates a genomic relationship matrix. You can also get relationship matrix for mixed models. You just need some sort of matrix that indicates the relationship between your samples. And this is just causing my computer to slow down a little bit, so GoToWebinar is complaining, but everything should be fine. There are a lot of things that we're not covering today. We do have fixation index, FST, by loci and by group. That is of interest to the agrigenomics community. We also have an inbreeding coefficient, and there are different ways that we can analyze the identity by state to look at this relationship structure. All of that we're not going to be covering today, but if you do want more information on that, just let us know and we can point you in the right direction, or we can set you up with a one-on-one webinar to showcase those features out. All right, now we have our results. And we'll go ahead and once again look at our XY scatter plot, or QQ plot. And add the y equals x line. And this is much better. If we enlarge this, the data, observed data follows the expected data until about 2.5, and then it starts to deviate. We still have significant results. It's not under the line. This is a much better treatment of the data with less inflation. But let's go ahead and get a number on that inflation factor. So again, on the p-value column, I'm going to select calculate pseudo lambda. Now this pseudo lambda value is slightly under 1, which would tell you that it would be underinflated. However, the QQ plot looks fine, and I would not be too concerned about this. Most of the time, most software would round this to 1 and call it good. If you were considerably lower than 1, then you might be concerned that you have adjusted the data to depress any signal. The last thing that we want to do is look at all of our Manhattan plots in the context of the genome, as well as look at our gene track for this data. So I'm going to go ahead and create a GenomeBrowse plot for this negative log 10 p-value. And actually, let's create a generic one. We'll go back to the Project Navigator, click on the GenomeBrowse window icon. And this is a dead track, so we'll go ahead and add. First, let's add in the protein gene predictions for Ovis Aries 3.1. Go ahead and plot that. And then we'll go back to our project and we'll add in our results. First, we'll add in the correlation trend test p-values. Then we'll add in the linear regression p-value and the mixed model p-value. And looks like I am running quite behind, so we'll go ahead and we'll just do this. And we need to jump out to the entire chromosome or entire genome. And we can color by chromosome. I'll just do that really quick on one of these. Get my controls here. And we can do that for all the same. We can easily scroll in. Let me just get this so we can see the genes. I'm just using my mouse wheel to zoom in this point of interest. And we can see that it's in a gene. We can click on this gene and in the data console jump out to learn more about it. Let's also look at the results themselves. We zoom back out. We can add a line. Let's add a line to the correlation trend test, since I know that that's the one that has its highest inflation value. So we'll click on the container itself and add light item. We'll color it red and set width of 2, put it at 6. Bonferroni correction for 50,000 markers at a p-value of 0.05 is negative log 10p is 6, so we'll just plot that. And we can see that, yes, there are quite a few markers that have a significant p-value under this test. If we were to do the same on the other two, we would see that there was just this one p-value for this test, the EMMA-X test, and just a couple for the linear regression with PCA correction. All right, so again, since we're running out of time, let's pop back to the demonstration portion. And before we move into the next demonstration, let's take time really quick for a quick poll. Let me just launch that. This should be fun. So go ahead and just take a few minutes to answer this poll. And then I will present the results. Golden Helix will be at PAG next week, and we will have lots of fun t-shirts. And we would like to encourage you to stop by if you are going to be there. And for those of you who can't make it to PAG, we would still be happy to send you a t-shirt if you want one. So just let us know, and we'll see what the rest of the attendees think. Let me just give you a few more seconds. And we still have a few people answering. All right, it looks like most people are done. So I'll go ahead and close the poll, and we'll share the results. All right. It looks like only a small number of you are going to be able to actually make it to PAG next week, but uh, a lot more would like a t-shirt, so just let us know and send us an email at info@goldenhelix.com and let us know your t-shirt size, and we'll be happy to get one sent out to you as soon as possible. All right, let's go ahead and get back into our next presentation. Okay, so now we're going to go to a DNA sequencing analysis workflow for agrigenomics. This workflow will be on 3 unrelated samples. We do have workflows for trios or small families. So for the DNA sequencing workflow, we are going to do the 3 unrelated samples, and the samples are from 3 different species, all aligned to the same reference genome. And our goal will be to look for putative heterozygous variants in a short list of candidate genes that are unique to one of the species. So let's talk about our data a little bit. I got this data from the Sequence Read archive, and there are 2 bovine partial exomes and 1 bison partial exome. All 3 are female. We converted the SRA data to FASTQs and then used GATK to convert them to BAMs and VCF files. We aligned them to the UMD 3.1 reference sequence. And this data was analyzed and referenced in the exome-wide DNA capture and next generation sequencing in domestic and wild species paper by Ted Cosart in 2011. And we'll kind of try to replicate their workflow a little bit. We do not have all of their data, so we'll do our best to give a quick look in the spirit of their analysis. So our workflow today will be a little bit of quality assurance. We'll talk about importing the data, calculate an alternate read ratio, do a little bit of read depth and quality, genotype quality filtering. We'll do a quick exon region filter just to make sure all of the reads are aligned to an actual exon region. We'll quickly look at some variant sharing between the samples, perform a variant classification on just the bison sample, and then we'll look at the list of candidate variants in conjunction with the VCF file and the BAM files, as well as other annotation sources. So moving right along, let's jump back over to our SVS project. We'll go ahead and save this and close out of this and open up our bovine data. Again, I've already imported my VCF files. To do that, we would just go to Import Sorted VCF. And I already have my VCF file compressed. If it's not compressed, you can just import a VCF file. The first step will be to compress it to make it run a little bit faster. It'll scan it. You can import a chromosome or a few chromosomes. You can split it by chromosome. If you've got whole genome data, it's a little bit better to do that for memory handling unless you have a lot of RAM available on your computer. I am going to only import those variants that pass filters. I imported the genotypes, the allelic depths, the read depths, and the genotype qualities. If you have multiple or 1 file per sample and multiple samples, all of this info data can also be imported as a spreadsheet. Since I have all of my data in 1 VCF file, it'll all come in as a marker map. But I'm not really interested in looking at these today. Any of the statistics that are output from your alignment algorithm will be included in the VCF file, and you can import those for analysis. So let's go ahead and cancel out of that. Our genotypes, we have about 258,000 variants for all the samples. And if a variant wasn't called for a particular sample, it's going to be filled in with missing. The allelic depth spreadsheet is a comma, each cell is comma separated. The first value is the number of reads for the reference, and the second or third and so on are the alternate read counts. And what we're going to do here is we're going to calculate a ratio of reads. In the DNA-seq menu, we'll go to Calculate Alt-Read Ratio, and it's just going to calculate the number of reads that are alt compared to ref, or total number of reads. And we can see that for these samples that do have a low number of counts, all the reads are alternates, and so the ratio is a value of 1. For this one where we had 1 reference and 3 alternate, the value is 0.75. Now, you can use these for filtering directly, or you can use this for analysis after you find your list of interesting variants. To get down to a small number of variants to look at, we will use it for filtering today. We'll go back to our genotype spreadsheet. And we'll go to DNA-seq filter based on VCF quality metrics. And we'll select those 3 spreadsheets: alt-read ratio, read depths, genotype qualities. And next. For alt-read ratio, which is the first tab, we need to be a little bit careful about how we filter. So I'm going to select advanced filtering options. I want to keep the ref genotype if the alt-read ratio is not 1. If the alt-read ratio is 1, then obviously it shouldn't be called the ref genotype. We'll keep ref alt genotype if the ratio is greater than 0.25, and we'll keep alt genotype if the ratio is greater than 0.5. For read depth, we'll keep the genotype if the integer value is greater than 10, and we'll keep the genotype we'll keep the genotype if genotype qualities is greater than 20. There are workflows on our website to talk about how to reach those numbers, or to come up with your own number based on your own data, but for the sake of time, I'm just going to go ahead and use those defaults, and we'll go ahead and say OK. All right. If the genotype or the read depth genotype quality fails those thresholds, the genotype will be replaced with missing. If an entire column happens to be filled with missing, that column will be inactivated and removed, so it will be removed from future analysis. And so right away we should see have good quality variants remaining for the 3 samples. A little bit more information: this top sample, the 7.8 is the bison, the 7.9 is the Bos indicus or zebu, and the bottom one is the Bos taurus, the domestic cow. And we are left with about 70,000 variants. So let's go ahead and create a subset spreadsheet. And from here, we want to look at how many variants are shared between the 3 different species. And to do that, I want to create subset spreadsheets for each species and only keep the columns that have an informative genotype or at least 1 alternate allele for each sample. So to do that, I'm going to go to DNA-seq, subset informative genotypes by category. I'm going to use the category as the row labels because I want 1 spreadsheet per sample. But you can do this as well for larger categories if you had a few samples of each breed. And we'll go ahead and say OK. All right, if we pop back to our project navigator, we're going to see that we have 3 spreadsheets here, 1 for each sample. Again, in the paper, they looked at heterozygote putative variants. So to do that, I'm just going to quickly drop down to only heterozygote calls. I can do that very easily using activate variants by sample genotypes. For each of these spreadsheets, I'm going to select alt-ref, heterozygote, and OK. And that drops down the number of variants considerably. We'll do the same thing for the other 2 samples. Oops. And lastly, all right, now we want to compare these 3 spreadsheets. So from the Project Navigator, I'm going to go to the Tools menu, compare variants across several spreadsheets, add those 3 spreadsheets. OK. And again, the top one is bison, middle one is zebu, and domestic cow. And we'll get a Venn diagram of all the data. We can see that 395 variants are shared between the bison, zebu, and domestic cow. That means they have at least 1 alternate allele at that location. The domestic cow has about 6 6,388 variants that are not shared with the other two. The bison has the most that are not shared, and the zebu has a little bit less variants that are not shared between the other two. I think that is mostly due to some of the quality scores we're missing for the zebu. All right. Now, this report also has the number of variants or counts that were used to create that Venn diagram. The next thing we want to do is look at our candidate gene list. In the paper, they had 203 candidate genes. I have a list of just 17, and I've got that saved in a DSF file. So we'll import that. We'll just quickly jump to the location. Short gene list. All right, and so again, the 17 candidate genes and a few different chromosomes. I am going to apply this gene list to our bison sample. We create a subset spreadsheet to begin with, and we'll go back to select, activate by gene list. And we'll select that candidate gene list, and we'll select the column containing the gene name. It could also be the row labels. And we'll select NCBI genes on the UMD 3.1 build. And we'll go ahead and say OK. That left 598 markers in the specified genes. So from here, we can go ahead and plot a variant map of those genes. What I'm really interested in is not so much these genes, but just how they are visualized in the BAM files. So I'm going to go ahead and hide that. We'll create the feature table for those interesting variants. We'll hide this. We'll add the BAM files and the VCF files. The VCF file will plot a variant map for all three samples. You can also plot read depths or genotype qualities or any of the other numeric scores in a plot. I'm going to go ahead and hide the pileups for right now and just have the coverage histograms. Okay, now this is the whole genome view, but I want to iterate through this list and look at my interesting variants. So I can click on one of the columns in the feature or rows in the feature list, and it'll jump to that variant. And I can use my down arrow to quickly flip through all of these candidate variants. And what I'm looking for is I'm looking to see if the histogram of calls matches what was reported in the VCF file. So this is a reported heterozygote call, and we can see that about half the calls are A and half are reference. Here we see a deletion that only has a read depth of 1, so that's not very believable. But we can go through and examine all of these variants. Because we did allow for 10 base pairs on either side of the exon, we skipped the exon filtering. So we're going to see some variants that are outside the exon regions. We could have filtered down to make sure that we had only variants that were within exon regions. And that is demonstrated in several of our other webcasts as well as tutorials. So again, we're just flipping through this. We're looking at the histograms of the coverage to make sure that the variant calls are match with what is reported in the VCF file. And here is a very good depth call. We've got 165 matches or reference. We do have 1 G and we have 78 T's for this location. So let's go ahead and plot the pileup for the BAM file. And we can see that we've got all of those alternate calls. We can split this pileup by strand, and we can see that this call is over both strands, and it isn't a gene. We can click on this gene again, just like with the sheet data set, and jump out and get more information about that gene. All right. I think we are at time, but we'll jump back to our slides really quick. And I hope those two demonstrations illustrate the power of SVS in advancing agrigenomics research. I know that they went through them really quick, but I hope that you're able to see that not just the SVS is good not just for GWAS, but also for DNA sequencing analysis and for agrigenomics data. SVS can also be used for copy number analysis and RNA sequence analysis with non-human genome or non-human data. And for more information about those workflows or other questions in general, please do not hesitate to email info@goldenhelix.com. And if you did want a t-shirt, please email your t-shirt size or t-shirt size for your spouse or your kid or your best friend. And if you do want to give SVS a try, request an evaluation at goldenhelix.com. And if you do want to try SVS 8, make sure that you note that. Otherwise, by default, you will get SVS 778, which is very similar but does not have GenomeBrowse embedded in it. All right. Now let's see how many questions that we have that we can answer today.
Thank you so much, Greta. It's so interesting to watch you walk through some of those common workflows for agrigenomic research. If you in the audience haven't already put your question into the questions pane, please go ahead and do so now. We'll stay on the line for just a few more minutes, maybe 10 or so, and answer just a couple of questions. And if we don't get to your questions, we'll go ahead and reach out to you after the webcast to make sure we answer anything you may have. As Greta mentioned, we do have a form on our website if you want to try out an evaluation of SVS. If you go to goldenhelix.com and go to the products drop-down and choose SNP Variation Suite, request evaluation is a button on kind of the top right. One more thing I did want to mention is that we are currently hosting a research competition, and that is open to both human and agrigenomic researchers. 3 winners will receive free copies of SVS, and the first place winner will also receive a new Dell laptop. We're accepting abstracts through the end of the month, January 31st, and more information on that is also available on our website. The first question we always have right off the bat is, yes, this webcast was recorded, and we will be putting it up on our website tomorrow along with the slides. Okay, so let's go over and answer a couple of questions that came in during the webcast. And the first one we have is, can we add any species to the software?
You can add any species as long as there is a genome information out there. So we either need to have chromosome sizes or scaffold sizes and number of scaffolds. Generally, we prefer diploid species just because that's what our software is designed for, but there are a lot of plant species out there that have a diploid equivalent. To make the software really useful, it's ideally that there's a gene track or some sort of preliminary gene track available and a reference sequence as well. As long as those criteria are met, then we can definitely add any of the species. We do have a tutorial on our website indicating how that can happen or how you can do that yourself. Most of our customers would rather have us do that for them, and we don't have any problem creating those reference assemblies for our customers.
And you said that was true for both SVS and GenomeBrowse?
Yes, whatever we create in SVS is the same information as used in GenomeBrowse.
Okay. Is there a step-by-step protocol on how to analyze data? Like, how do I know which analysis I should use so that the data comes out to be as accurate as possible?
We have a lot of tutorials on our website, and most of our agrigenomic customers are a little bit concerned because they're human-based. Most of the workflows are the same. The only limitation is sometimes we do not have some of the annotation sources that are available for humans available for non-human species. So those steps would just need to be skipped. For GWAS workflows, it's pretty much all the same. It doesn't really matter where the data comes from. The steps are the same. For sequencing workflows, again, the filtering, there isn't as many sources available for filtering, and so anything that doesn't have a non-human equivalent or your species equivalent, you just have to skip that step. Most of the non-human species filtering, it would be just getting down to exon regions or regions of interest and then performing variant classification, which is another thing that I didn't get a chance to go over today, but variant classification is very easy to do in SVS, and it is a good way to get down to non-synonymous variants, and it doesn't, all you need for that is, again, a gene track and a reference sequence. You don't need to have any functional prediction databases to do variant classification.
Okay. And if I have questions about the tutorials or what to do for my agrigenomic research versus a human or with the software, what should I do?
Just email info@goldenhelix.com or support@goldenhelix.com, and we can either set up a one-on-one webinar or answer your questions about the tutorials or your workflows via email as well.
Okay. Looking at the next, what kind of computer hardware did you run this webcast on in terms of number of CPU cores, amount of RAM, etc.?
I do have a nice amount of RAM on my computer. I do have 24 gigabytes of RAM, but I wasn't getting close to the limitations of that in anything that I was doing today. GoToWebinar just has a tendency to want to use up a lot of RAM, and so when you're running an analysis, it tends to complain even though it's not going to shut down your computer. And I do have, it's a 4-core machine.
Okay.
So not anything too fancy, but not a laptop either.
Okay. I think we have time for just one more question. Is the data that you use today available for the audience to analyze themselves?
Well, the sheep HapMap data is available if you request it from the consortium, and so I'm not authorized to re-release that, but if you contact them, then they will provide some information about what you're going to use the data for. They wouldn't have any problem, I'm sure, releasing it. The phenotype I could release since it is something that I created myself. The DNA sequencing data that I used is, again, publicly available from the Sequencing Read Archive on NCBI's website. And we can have, we're going to have a short little paper talking about how we got that data and how we processed it to get it ready for analysis. So we can't, we would have to look into whether or not we can provide that directly, but we can also just point you in the direction and talk you through how we created that data so that you could get it yourself.
Okay. Thank you again so much, Greta, for, like I said, the great webcast, and thank you all for attending and for submitting your questions. If you are at PAG next week in San Diego, I hope you stop by and say hello. I would love to, I will actually be there this year, so I'd love to talk to you and see what you're doing with your research. Have a great Wednesday, and we'll see you all again soon.
Thank you.